Let's discuss the implantation today. Definition: implantation is the process by which an embryo attaches to the uterine wall and penetrates first the epithelium and then the circulatory system of the mother to form the placenta. It is a process that is limited in both time and space. The embryo in the morula stage enters the uterine cavity where it continues to divide to form blastocyst. This happens in a defined time period of between 72 to 96 hours after fertilization. Successful implantation depends on three factors: embryo quality, endometrial state of receptivity, and synchronized dialogue between maternal tissue and the blastocyst. Preparation of implantation. The primary endocrine requirement for implantation to happen is the presence of progesterone. Endometrial receptivity is heralded by progesterone induced formation of pinopodes. The window of implantation or the window of endometrial receptivity is from day 20 to day 24 of a 28-day menstrual cycle. A dialogue between endometrium and early embryo involves multiple factors. The first and most important is early pregnancy factor. It is detected in maternal circulation within 1 to 2 days after fertilization. Prior to implantation, early pregnancy factor is produced by the ovary in response to a signal from the embryo. After implantation, EPF is derived from embryo and has immunosuppressive properties. Second is HCG, human chorionic gonadotropin. It is secreted by blastocyst. It starts 7 to 8 days after fertilization. It enhances steroid secretion from corpus luteum. Prostaglandin E2, it is secreted by secretory endometrial epithelial cells. Synthesis is increased at the implantation site. Coming on to stages of implantation, the human blastocyst remains in the uterine secretions for approximately 1 to 3 days and then hatches from its zona pellucida in preparation for attachment. We can see in the picture the early blast, then the late blast with the expanded cavity. As the cavity keeps on expanding, it becomes expanded blast. Finally, in the fourth and the fifth picture, we can see a fully hatched embryo. The implantation site usually lies in the upper posterior wall in mid-sagittal plane. So the first stage of implantation is apposition. The connection between blastocyst and endometrium is maintained during apposition. While the second stage is adhesion, the trophoblast adheres to the endometrium strongly and starts penetrating it. Final stage of implantation is invasion. The invasive blastocyst penetrates through the luminal epithelium. The endometrium produces at least 3 cytokines involved in implantation: colony stimulating factor 1, leukemia inhibitory factor, and interleukin 1. Subsequent events. Invasion of the trophoblast happens via degradation of extracellular matrix. 3 subsequent interactions occur. Trophoblasts intrude between the uterine epithelial cells. The epithelial cells are lifted off the basement membrane. The trophoblasts can interdigitate underneath. Finally, fusion of trophoblast with the uterine epithelial cells. Vascular changes during implantation. Uterine spiral arterioles are invaded by cytotrophoblast. Maternal endothelium is replaced by cytotrophoblast tissue as far as first third of myometrium. This replacement may be governed by selectin family of surface molecules. Matrix metalloproteinases play an important role in implantation. They are key players in matrix degradation during the trophoblast invasion. Collagenases, gelatinases, stromolysis, these are a few matrix metalloproteinases which play a role in implantation. Next is the implantation window. Specific period of time during which implantation is possible is the implantation window. It varies from day 20 to day 24 of 28-day menstrual cycle. Pinopods. They are bleb-like cytoplasmic protrusions found on the apical surface of endometrial epithelium. Pinopods are several micrometers wide and project into uterine lumen above the microvilli level. Electron microscopy is the major tool used to observe these structures. Blastocyst attachment has been shown to occur onto the top of endometrial pinopods, and they are dependent on progesterones and homeobox gene 10, whose expression is necessary for endometrial receptivity to blastocyst implantation. Functions of pinopod: they protect the blastocyst from being swept by cilia on the endometrial wall, and they endocytose uterine fluid and takes the wall closer to the trophoblast floating in it. A pictorial image of physiology of implantation. We can see here the blastocyst along with the trophoblast and the inner cell mass. In the inlet you can see the pinopods which are in green. The red one is the mucin, and there are L-selectin ligands with and L-selectin, endometrial stromal cell, decidual endometrial stromal cell, macrophages, uterine NK cells, and leukemia inhibitory factor 1. All have been shown. First step of apposition followed by adhesion, then invasion or penetration, and finally the decidualization has been shown. Finally, little bit about immunology. The pregnancy, the conception, is associated with Th2 T helper cell 2 dominance. So, the interleukin 4, interleukin 6 and interleukin 10 inhibit Th1 cell induced tissue factor by monocytes, and the predominance is associated with pregnancy, while excess of Th1, it is associated with the rejection of embryos. Thank you everyone for the patient listening of this basic topic. So we are continuing with the basics in reproductive medicine, and I hope these topics are clear to you. If any doubts, please tag me in the comments, and if you want any other topic in the basics of reproductive medicine, please put it in the comments so that I can make video regarding that, and so that it can be helpful to all the students who are reading and who are interested in reproductive medicine. Thank you.